So are we set?
Yeah, we are ready. I muted
Okay.
everybody.
Okay. So yeah, time up. I want to welcome everyone to today's presentation. It has been long awaited, and we are so grateful that we are seeing today. So without further wasting of time, I want to appreciate everyone that is on call right now, starting from our interns. I really want to appreciate you. HackBio love you, and we are grateful for all you've been able to do. Um, if we are to go back to the genesis of how we started as a team and how we had you guys on board, we had over 780 people who applied for this, and today we are just left with less than 90. And so you should consider yourself lucky of being part of this final presentation this evening. Without wasting your time, the programs are already set. You must have been told there are judges that will listen to your presentation and will score you, and then we have a winner. In as much as it was an internship for everybody, we want to appreciate some very talented group that really gave in their all into it. So apart from the skills you must have achieved and you must have gotten, we would award the best group and some of the individuals that did excellently well in the course of the internship. Um, okay, I'll go further now to announce to us the judges so that we can start as fast as possible. So with us now, we have a very big friend, a very big friend to the association, who has been of great support since we started, in the person of Mr. Oluwale Gabriel. We also have our very wonderful sponsor, Ms. Alice, who have joined us already, and these people will form the group for the judges. So let me go to proper introduction of the judges now. Mr. Afolayan Oluwale Gabriel is the co-founder and secretary, Board of Trustees, CMSBAP. He has over 12 years exciting career working for non-profit and government institutions. He currently leads the effort to establish the Nigeria Biobank, which will support understanding the genetic marker of diseases and treatment outside of diseases and treatment outcome in Nigeria. I beg your pardon. As research secretary, Afolayan ensures that evidence from research inform policy decision in the campaign against NTDs, neglected tropical diseases, at the Federal Ministry of Health, Nigeria. You're welcome, sir. Also, we have Alice. Alice is the CEO and founder of DNA Compass. DNA Compass is a California benefit corporation wanting to facilitate the use of ESRI's ArcGIS infrastructure to distribute maps of COVID-19 genomic markers and to help entities stratify population by genomic risk. Currently, the DNA Compass has been used successfully in the fight against rare diseases, cancers, and more recently, COVID-19. We're also expecting one of the judges in person of Dr. Kayode Ahmed, who is a researcher from Harvard Medical School. So I'll move on now to the brief overview before the presentation by Wale. Over to you, Wale.
Thank you, everybody, for joining us on this call. I sincerely appreciate the presence of our sponsors and our partners, including Simesbaf and DNA Compass, the person of uh, Mr. Gabriel Afolayan and Alice Radton from DNA Compass. So my talk will be very little because we have very little time. So I am very happy to have everybody here for the, let's say, the final time, and accept my congratulations on the success. So we started with 700 interns, and then right now we have uh, less than 80 interns with that have successfully honed their skills in bioinformatics. So it is interesting to also know that all of them, we had a mix of experts. We also had a mix. We also had intermediates as well as basic learners. We also had novices that were able to make it to the end. Interestingly, everybody, the workspace was collaborative enough, and everybody was able to grab interesting skills in bioinformatics for development of goals, for development of solutions, health-based solutions, and for the betterment of humanity. So throughout this internship, we were able to produce five major publications, which five major manuscripts, which will be submitted to journals, international journals of high repute. And then we also had collaboration with the African Galaxy Network, which is just starting out, but then we already joined the team. Some of the genomics and drug development interest persons have joined the Galaxy Network from HackBio. Also, we have the vaccine guys, the guys that are working on vaccine. They were able to deposit optimized variants for the novel coronavirus. That's the most important issue right now all over the world. Also, the drug development guys were able to identify and deposit about 500 phytochemicals from plant source that were uh, potential drug drug candidates for COVID-19. Also, we also have a strong, unique collaboration with DNA Compass, and we are collaborating with them on the level of a genomic infrastructure for global monitoring of rare diseases um, and COVID-19. Also, we already have one of the important things we also got from this internship is a resilient bioinformatics community, and because of the high level of collaboration and connectivity between the interns, we have decided not to close down the workspace after now. So the workspace lives on, and the community lives. We can continue to uh, network, carry out research, virtual laboratories, virtual startups that can immediately start on Slack as well. So that would be all from me for now. I would enjoy everybody to sit down as we enjoy the presentation from the interns. So our mentors, I believe we already have access to these sheets, the spreadsheets, and then we can immediately begin to assess once interns begin to present. So this is the way the presentation will go. Interns they have created a non-technical guide, like it's a video abstract. So they will share a non-technical guide that even a dummy should be able to understand. We don't have dummies anyway, but a novice, a non-technical person in, let's say, bioinformatics, will be able to understand and appreciate what they have done. So in this way, also the presentation would be a little bit technical, but then they will try as much as possible to show to show us the relevance to global human health and the current efforts towards COVID-19. So thank you everyone once again. Let the presentations at the next thing the agenda should continue. Samson, over to you.
Thank you so much, Wale, for that brief overview of our achievements so far since the beginning of the internship. It might interest you to know that it has been a 5 week of internship, and the work these guys are having is awesome because at the end of the day, for the judges, you are going to be wowed by what you will see. We are really, really, really happy that this internship has been of great importance and benefit for so many people. So we'll move on to the next on our agenda, which will be the future prospects by JK.
So I will unmute him now. Sorry.
Okay.
Hello, everyone. Can you hear me?
Yes, we can hear you.
Oh, I'm glad to be here. It was actually nice doing this with everyone in the past 5 weeks, working on bioinformatics, working on data science, working on things that are actually important and crucial to science and to our existence. And it's a good thing. Future prospects going forward, we'll be keeping the community active. Essentially, we need a community for us to have a platform where people can interact, they can work together, they can keep things running. So, because from communities, uh, startups, companies, businesses will be formed, research groups will be formed, and from there we keep that spirit of collaboration on, and we keep working together. From there, well, we can't say exactly what will happen in the next 10, 15 years, but hopefully we can make some clear predictions that with our community, maintaining a stable community and working hard towards keeping the community active. From there, we can actually have a group of people who will spread the impact into the other parts of the scientific community, of the science community. And when the impact spreads all through, then we begin to see the changes that we desire in science. Just as we have seen in software development, as we saw people do with open technology, open source, and all those. I think we can begin to have that from now. We have a lot of GitHub repositories that we already formed, a lot of works that we've made public from HackBio. So going forward, we'll be having more of those, and we look into the suggestions we had from the interns and the mentors all through the program. Apart from that, uh, we'll be creating an initial program that will serve as a prerequisite for people who have no knowledge about bioinformatics before coming to the next internship, HackBio internship. So we'll be having that program too. I think that's the basics that we have for now. Thank you.
Yeah, thank you very much. I think Samson, maybe you shouldn't unmute. You shouldn't mute yourself again. Once you are mute, I always have to unmute you again.
I'm so sorry about that. Yes. So you heard from JK. I think Wale has 1 or 2 things to add to that before we get to the presentation.
Okay, I don't really have anything to add. We also wanted judges to say a few things. maybe just say hi to the interns, and then the presentation can start.
Okay.
I'm going to
Okay.
unmute them now.
So can you unmute the judges now?
So, Mr. Gabriel, I just unmuted you. Can you?
Okay. Good evening, afternoon, depending on the part of the world you're joining the program for. Let me first congratulate once again everyone that made it to this final stage, most especially the HackBio team for this wonderful initiative. Hearing the outcome of the 5 weeks intensive internship program, I'm excited that 5 manuscripts came out from these 5 weeks effort. That's quite interesting and highly commendable. Let me say to everyone making the presentation this evening that we're going to be very friendly, and we hope that irrespective of the outcome of the presentation, please keep in mind that you are all winners. You're fantastic individual, and we look forward in the nearest future of your great contribution to the global, effort to curb these diseases that are ravaging the world. So congratulations once again to everyone, and I look forward to an interesting and insightful presentation. Thank you so much.
Thank you, sir.
Alice, I think I unmuted you. Yeah.
Yes, I'm just, thank you for the chance to see these presentations. The topics are excellent. I'm very much looking forward to it. And with that, I don't want to take up any more time. I can't wait to see the talks.
All right, thank you so much for that, Alice. We also cannot wait to hear from these interns as well. So we'll move next to the presentation proper now. But before we start, I want to say one or two things before the presentation starts. So this is clearly to the interns. There are criteria, there are points for most of the things you are going to be doing on while presenting. So you have to be very, very mindful of some of the things I'm going to call out, because you'll be graded based on this. Your clarity, your organization, your content, your creativity, your timeliness, then your ability to answer questions. Ability to answer questions. Ability to answer questions. I repeated that twice so that you know that there's emphasis there. You must be clear while answering questions. And this is how it will go from drug development 1 to drug development 2 to genomics 2, then genomics 1, then proteomics and vaccine as the 5th group that will present. I wish you all the best. Your time starts now. Drug development 1, you are on.
So drug development 1 is taken by Suritra, I believe. So I would unmute you ahead of time so you can start preparing your slide while I display the video for everybody. If you know your background will be noisy, maybe you should just mute. So I will start sharing my screen.
evaluated in this study. We employed a virtual screening of phytochemicals from 19 different herbs and medicinal plants, ranging from compound classes of flavonoids, coumarins, terpenoids, polyphenolics, saponins, thiophene to alkaloids. We also performed a structure and sequence comparison of SARS-2 NSP9 with that of SARS and HCOV NSP9, which reveals that SARS-2 NSP9 is more closely related to SARS. This finding can be further utilized for the design of broad-spectrum inhibitors for the prevention of host cell attack. Extensive screening of the manually curated library of 521 molecules via docking threshold scores and stringent ADMET filters identified 5 leads for non-structural protein 9, 23 phytochemical leads for purine, 18 leads for ORF3A, and 19 leads for interleukin 6. Among the top 5 leads, ocnaflavone, a bioflavonoid extract from Lonicera japonica, commonly called as Japanese honeysuckle, and licoflavone B, a flavonoid extract from Glycyrrhiza glabra, commonly called as licorice, showed promising polypharmacological prospects as identified by the docking scores, ADMET properties, and binding interactions with the receptor site residues. Simulation studies were performed for the top-scoring NSP9 ligand complexes to assess the stability and protein-ligand contacts, and binding energies were calculated employing the MMGBASA method. The current study comes up with convincing evidence about antiviral and anti-inflammatory indications of the identified phytochemicals with huge potential to inhibit viral entry and replication as well as disease progression simultaneously, further opening up the quest for in vitro and in vivo experimental study.
Okay, so I'm going to stop sharing and then I will allow you to share your screen. So Suritra, please go ahead.
Yeah. Am I audible to everyone?
Yeah, you're audible. You're audible.
Yes, you are.
Okay. Thank you so much, everyone. Hello and good evening to all who have joined in this evening or afternoon as it applies. This is Suritra here that you have been seeing behind the screen till date, and I feel extremely honored to be presenting the work that our team, Drug Development One, have executed in our tenure at HackBio. The video pitch already gave a mini overview of our work right from the start to the end, but let's talk a little bit about why virtual screening. We are all aware how COVID-19 has become the talk of the town, and urgent remedies of management of public health and economy are the needs of the hour. Conventional experimental methods like high throughput screening continue to be the best method for rapid identification of drug leads through extensive individual biochemical assays. However, the huge cost and time expenses has led to the integration of cheaper and effective computational methodology, namely virtual screening. It's a computational screening method which is widely applied to screen in silico collection of compound libraries to separate the non-binders from the binders. However, both methods come with their own set of inherent challenges and limitations. So a manually curated library of 521 phytochemicals from 19 different medicinal plants with proven antiviral and anti-inflammatory properties was generated by literature survey. And we have stored it in the MQ web server repository. So we performed So we virtually screened the library using MCool web server against 4 targets that we chose. NSP9, which is an RNA binding protein and has an extremely important role in the viral replication process. ORF3A, that plays a major role in inducing cell apoptosis and cellular inflammation. Furin, that serves as the storage of pre-glycoprotein processing of spike protein and facilitates the viral entry to host cells and intercellular invasion. And interleukin 6, an inflammatory cytokine, especially for the ICU COVID-19 patients owing to its role in the cytokine storm. Now, NSP9 is one of the proteins whose biological function in the replication machinery is still uncharacterized. Structural comparison with other related virus strains can give us information for designing a broad spectrum inhibitors. So we perform a protein blast and multiple sequence alignment, and the results demonstrated a sequence percentage identity of 97.35% to that of SARS-CoV NSP9 and 44.25 for NSP9 of human coronavirus. We then superimposed the NSP9 structures of the two viruses using PyMol functionalities, and using the executive RMSD values and match align scores, we confirmed that SARS-2 NSP9 is more similarly structured to SARS NSP9. We started with the library of 521 ligands and allowed all those molecules to pass through that have 2 or lesser violations of Lipinski rule of 5. This allowed us to test for a broader chemical space, and this was also necessary since traditional antivirals and natural product-based antivirals usually violate these rules. We received 452 molecules, and Vena docking was performed to receive the top 30 hits for each of the targets. In the next step, we employed a stringent docking score benchmarks to allow only those hits that had more binding affinity than that of the standard drug molecules used. And the results that we got are 6 for NSP9, 23 for RF3A, 30 each for furin and interleukin 6. Next, the potential hits were subjected to ADMET filters of carcinogenicity, phosphoglycoprotein substrate, AMAC mutagenicity, and cytochrome P450 metabolism. At this point, we have our lead candidates. Among all the leads identified, we observed that leucoflavone B, ocnaflavone, and hispaglabridin were the 3 out of the 6 for NSP9 that possess polypharmacological properties and have excellent binding affinity values for all the receptors. However, interestingly, hispaglabridin B failed at the ADMET filter, and hence this opens up prospects for lead optimization studies. From here on, we zoned down our study to the protein target NSP9 due to its essential role in viral progression and due to computational limitations. Binding interactions were studied, and we used the MMGVSA binding free energy to inform us about the complexes that we would further carry forward for MD simulation studies. To keep it brief, we performed a 100 nanosecond molecular dynamic simulation assisted by our MENTA to analyze the stability of 3 top scoring complexes, and found that NSP ligand complexes of leucoflavone B and ocnaflavone had stable and consistent contacts with specific amino acid residues throughout the entire duration with minor fluctuations, unlike hispaglabridin B that didn't have specificity. This further strengthened our finding of the 2 drug candidates for their potential inhibitory effects against the multiple receptors under scrutiny. And also of interest were the surface distribution of hydrogen bond donor acceptor sites, hydrophobicity, solvent accessible regions, and aromaticity for understanding the binding interactions even better. The COVID-19 pandemic has become a major public health issue of global concern. Now, as I present this slide, the total count of infection stands at 26.7 million, and the death toll stands at 880K. And now it is much more than just a health crisis. The development of a potential drug candidate for new targets and using natural phytochemicals for the treatment of this infection will definitely be a breakthrough in the scientific community that will benefit each and every affected societal domains. To sum it up, we see SARS-2 NSP9 as structurally more similar to NSP9 of SARS than human coronavirus. We identified 5 lead candidates for NSP9, 18 for ORF3A, 23 for furin, 19 for interleukin 6. We assessed the excellent polypharmacological properties of ocnaflavone and leucoflavone B validated by simulation studies, and thereby this study serves as an initiation for the preclinical sets of experimental verification. Last but not the least, on behalf of each and every member of the team, we wish our heartfelt thanks to the entire platform of HackBio team and all the associated sponsors and partners who have proved us that even this is possible and have been successful in making HackBio a benchmark in the history of bioinformatics. Special thanks to Neve, our mentor, for his guidance. And thank you so much, everyone, for your patient hearing.
Okay. Samson?
Yes, thank you so much for that presentation. Drug development 1. It was a very, very nice presentation. Now we move to question and answer. Over to our judges. Any question for the drug development 1 group?
Hi, this is Al.
So if there's any question.
Yes, someone was
It's,
speaking. Okay, okay. I
yeah,
think
it's
Alice is
Alice.
speaking.
And I was going to ask if you had resources, what would be your focus? What would you like to, what question would you like to pursue next?
Okay, so since we identified leucoflavin B and ocnaflavone as having polypharmacological properties, we would definitely want to have a verification by in vitro tests. And yeah, and if
If
we
you
get good results, and then we can move forward with it
if you had to pick
accordingly.
one, which one would you pick?
I would pick leucoflavone B because the MD simulation results that we saw uh, much better than ocnaflavone.
Very good, thank you.
Thank you so much.
Okay, Mr. Gabriel, do you have any question?
Um, I'm just trying to ask about the interleukin, if I hear the presenter correctly.
Yeah.
So what happened after they had met properties for the first 4 virtual hits that you identify? What happened to interleukin 6? It's like I didn't pay attention. You only major on NSP9. So what happened to the
Okay.
interleukin
Okay.
6?
Okay.
Yeah.
Yeah. So we since we had less time and computational studies are usually very time expensive, we just chose for the time being, we chose and we zoned it down to NSP9 because of its important role in the viral replication. And if given a chance, we would definitely also like to perform it for all the 3 other receptors that we have chosen. So that will definitely give a better, like substantial support to the already established results that.
Evidence. Oh, okay. Yeah. Thank you.
Thank you so much.
Thank you so much, Drug Development One. Thank you for that beautiful presentation and the way you answered your question. We will now move to Drug Development Two.
Okay, so so Rachel, kindly stop your presentation so I can, yeah, thank you. So I can share screen now.
Hi, I am Annapurna. I am here on behalf of.
So this is Drug Development 2 team, and here is their brief
Hi, I am Annapurna.
introduction.
I am here on behalf of my team to give you an overview of our project. Coronavirus is continuing to spread across the world with more than 25 million confirmed cases in 188 countries. More than 840,000 people have lost their lives. It spreads from the infected person through the respiratory droplets by coughing, sneezing, etc. Approximately, these droplets can stay in the air up to half an hour depending on the size. When these droplets are inhaled, the virus settles in the nasal passage and starts replicating. It takes over the healthy respiratory cells. Our lungs have tiny air sacs called alveoli that takes up oxygen and keeps our body going. The virus settles in the air sacs, causing shortness of breath. Researchers around the world have been working hard to find a vaccine for COVID-19. Drug repurposing is one of the methods to find the cure faster. It involves investigation of existing drugs for new therapeutic properties. Our project focuses on FDA-approved drug candidates and 6 potential protein targets in coronavirus. The targets are nothing but proteins or enzymes that is involved in causing the disease. We performed virtual screening with nearly 5,000 FDA-approved drug candidates. The best from the 5,000 compounds were then compared with standards. ADMET analysis were then performed. ADMET stands for absorption, distribution, metabolism, excretion, and toxicity. Absorption is the amount of drug that is available for the target cells. The drug has to be carried to its target site, often by blood, is called distribution. Compounds are broken down as soon as they enter the body. So the drug we take is broken down to smaller compounds called metabolites that may or may not be active. This is called metabolism. The metabolites need to be removed via excretion, usually done by the kidney. If compounds are not excreted properly, the compound might cause damage to the organ or the organism. This is called toxicity. Thank you.
Okay, so I'll stop sharing and then you can start sharing. You can start your presentation immediately. The time has started. Okay, probably I have to unmute you as well. Pardon me, I'm going to unmute you. Yeah. Your time has started. You are not to present, you are not to share.
Your time is counting. Drug development too.
Good evening, everybody. Am I audible? Hello.
Yeah.
Yes, you are.
Hello. Good evening, everybody.
Good evening. Everybody has been muted. Just go ahead, please.
Yeah, my project is about drug repurposing strategy of FDA-approved drugs against SARS-CoV and in silico study. So we introduced as the coronavirus disease pandemic that is caused by the coronavirus has been ranging throughout the globe, throughout the world. It has been affected by 188 countries, and it has more than 26 million cases and over so many deaths. So we focused on the project. Our project mainly focuses on drug development for the coronavirus, and we have seen here that a traditional drug development can take up to 1 year or more. So we have chosen this method of drug repurposing that has proven to find vaccine with a faster and a cheaper alternative. So we can see that drug development usually takes from the clinical studies to that has 25 to 30% of success rate. The candidates from that pass phase 3 are from 25 to 30%. But while using drug repurposing and other bioinformatic methods, we can go for more success rate. And for the materials and methods, we first identified and prepared FDA-approved drugs. That is up to 5,334 drug candidates from Zinc database, PubChem, Drug Bank, etc. And we prepared the drug library. And after that, we prepared the protein ligands, and the docking was done using the Pyrex software. And after the docking results, we chose actually 6 protein targets that include the replicase polymerase and RNA dependent RNA polymerase and ADP ribose phosphatase and NSP9 RNA binding protein and 2 other proteins. And molecular docking was done for all the proteins with the drug candidates. And we selected some of the best candidates ligands using the binding affinity, and we compared them using the standards. So the docking results of the 6 protein targets were then compared with the standard drugs, that is a hydroxychloroquine, lopinavir, and redismivir. And the 10 ligands that were chosen had for each protein had lower binding energy than the standards. And the common ligands for all the 6 proteins are mentioned here, that involves doxycycline, ratiglivir, and dixoxin, and piroxicam. And these are the results. We can see that we have dogged for the standards, that are hydroxychloroquine and lopinavir and redismere. We can see the 6 target proteins here, and what is the values for everyone. They differ for each protein. So we took this as a standard and compared them with the results that we got. And we observed our results in Discovery Studio, where we could see the interactions between the protein and the ligand. I have mentioned 2 of the ligands here, which shows the pi-pi bonds, the van der Waals interactions, hydrogen bonds, and the favorable and the unfavorable bonds. And here are the results of ADME analysis that we did using Swiss ADME. And I have placed here the 6 best ADME results that we got from all the 10 for each protein, and I've placed only some of them here because we couldn't place everything. And we can see that some of these ligands are totally follow the rule of 5, Lupinski's rule of 5. And we know that currently COVID has no approved drug yet, and it still spreads around the world. And repurposing existing medicines that we have focused on drug targets is likely to offer more rapid vaccine for the COVID. And the results that we have got, and we would like to conclude telling that catmidip and Rattlegiver has a good affinity and good ADME results that follow all the drug likeness rules, including the Lupinski's. And we can see that all the different proteins have different ligand that has good binding energy, but catnip and Rattlegiver has the best for all the proteins. It's common for all the protein targets we have selected. And lastly, I would like to thank HackBio and our mentors, and also all the sponsors and the partners. Thank you.
Thank you so much, Group 2, Drug Development, for that wonderful presentation. Over to the judges. Questions for them?
Yeah, you have been unmuted.
Questions from the judges?
Is there, given that sorry, I'm having trouble formulating it. I'm curious if, it seems like you took a very similar approach to our first presentation, yet came up with a different list, quite a different list. Just wanted to get your thoughts on why your list is different, and also possibly the benefits of the approach that you've taken.
Okay. First of all, the team 1, they have done with protein targets. I mean, the ligands that are mostly from plant source, and we have not chosen plant source. That's why we have different results. And I would like to also tell that the methods we have chosen is totally different from them, because they have chosen plant samples and chosen the drugs from those plant targets, but we have chosen FDA approved drugs directly from the library. So
then the efficacy of these drugs and the safety would already been established, and so you were expecting a more rapid adoption, in effect.
Yes.
That does seem like a good approach. And what, if you had to pick one drug, what was the one that you would advocate for pursuing?
We would like to take Rattlegiver, because it shows better results with all the proteins. The other one also shows better, but Rattlegiver has a better ADME approach.
Thank you.
Thank you, ma'am.
Thank you, Alice. Mr. Gabriel?
Hello, sir, Mr. Gabriel.
I think he's mute, sorry.
Okay, please unmute him.
Yeah, I've done that.
Okay, I had a network failure at a point, but can you just go back to your...
So can you share your screen again, Anna?
Yes, sir. Which slide to be specific, sir?
9 and 10.
Yes, sir. Hello?
I think there's a problem with the audio device on the other end. Okay, maybe you can keep the question. We will probably address it.
Okay.
Yeah.
Okay. So group 2, drug development, you may likely have a question when we have Mr. Gabriel join us.
Okay,
So
sir.
thank you for the beautiful thank you for the beautiful presentation and the question and answering. It was wonderful. Without wasting
Thank you so
much
much, sir.
time, let's move on. You're welcome. Without wasting much time, let's move on to the next group, Genomics 2. I hope you are set.
Repeat it by.
Genomics 2, your time starts now. Genomics 2. Genomics 2. Oh, your pitch. Oh, Tati, we lost Wale there. Adewale will connect soon. Please just give us few minutes to have him connect. Then yes, give us few minutes. I think he's not online. Some technical problems there. You will come online and play your pitch immediately. Genomics 2, you may have to start your presentation without your pitch for now. Please bear with us. I don't think it will affect your scoring, please. He had a technical issue. Oh my God. Okay. Okay, Anapora, you appear as the host. Can you unmute Winifred? Anapona, can you hear me? Anapona, can you unmute Winifred?
Yes, sir.
Please unmute Winifred.
So I have not muted anybody.
No, you appear to be the host now. You have to unmute Winifred, please.
Sir.
Hello, can you hear me? Or you can make me the host.
Yes, sir. 1 second, I'm trying.
Okay, please unmute Winifred.
So whom am I supposed to?
Winifred.
Okay, thank you very much.
You're welcome.
Can I start?
Yes, but I want to apologize. Wale will join us shortly. He had some technical issue. So for your pitch, we will consider that. Please, we are so sorry about that. So you can start now.
Okay. Hi, everyone. We are Genomics 2. We are presenting on transcriptomic dysregulations associated with SARS coronavirus 2 infection in a human nasopharyngeal and peripheral blood mononuclear cells. More than 24 million people have been infected with the SARS coronavirus 2, and more than 820,000 deaths have been recorded globally. And on the right side, I show a cartoon diagram of the global map, and the bigger the dot in red, it shows a higher number of cases recorded. Angiotensin converting enzyme 2 receptor gates the entry of SARS into the host cell. Transcriptomic profiling has been used in evaluation of gene expression profiles in response to disease or treatment. However, molecular mechanisms that underlie pathogenesis of COVID-19 are still not clear. And therefore, our study aimed to understand host response to SARS coronavirus 2 infection from two distinct sample types. Our objectives were to determine differentially expressed genes and identify prominent immune response genes associated with SARS coronavirus 2 infection. Our approach was to download read count data from Gene Expression Omnibus database in NCBI under the accession numbers shown here. And we did a differential gene expression analysis using DSEC2 package in R. And we went ahead to do enrichment analysis in gProfiler using gGhost tool. Here I show a bar plot of the number of samples. On the y-axis is the number of samples, and on the x-axis is the sample types. And we can see here is that what is in blue, these are the healthy individuals, and what is in red, these are the COVID-19 patients. We did a principal component analysis to understand the variability of our data. And from what you can see in the first principal component analysis here is that the cyan blue dots denote the healthy controls, while the one in pink denote the positive patients. And for the nasopharyngeal samples, as you can see, there is a significant overlap between the healthy and the COVID-19 patients. With the PBMC samples, we can see two distinct clusters, that is the positive and the negative, all the healthy controls. On the far right, I show volcano plots. The y-axis is the negative log of the p-adjusted values, and the x-axis is the log 2-fold change. Every dot in the volcano plot is a gene, and those are the genes that are associated with the that are in red are upregulated, downregulated. Ones, they were not differentially expressed. Interestingly, as you can see, we've got a very high number of upregulated genes in the PBMC samples when in comparison to the downregulated genes. We went ahead to construct heat maps for the top 20 genes. And just to point out here is that the yellow bar represents the positive samples and the cyan blue denotes the healthy controls. And the scale for these heat maps is that the hotter the color, then it denotes a relative increase in expression. And as the color dims down to green, then it shows a down regulation or a relative decrease in expression. Interestingly is that with the nasopharyngeal samples, we can see heterogeneity in the gene expression profile. While with the PBMC samples, we can see some distinct pattern between the positive and the healthy controls. We identified 13 commonly regulated genes between the two sample groups, and we did hierarchical clustering, and we were able to discriminate between the PBMC and the nasopharyngeal samples. We went ahead to really understand what the biological relevance of the differentially expressed genes. And here we show the top ones that are in red are the immune functions, while those that are in blue are the metabolic functions. And just to note is that with the nasopharyngeal, we can see the inflammatory response. We can see chemokine and cytokine activity. We can see leukocyte migration. Yet is PBMC samples, we can see classical pathways. We can see complement cascade, the B-cell mediated immunity. And we went ahead to model the mechanism. Is a cartoon of an invading virus. Then it releases the viral proteins that are bound by the antigen presenting cells, then leading to production of cytokines that then recruit the leukocyte into the site of infection. That's on the nasopharyngeal cells. Then with the PBMCs, it's a cartoon of the B cells. They mediate production of antibodies that bind to the virus. Then it recruits the classical pathways, as you can see, the C1, C2, and C4, and then C. And with the nasopharyngeal cells, we can see cellular response was more predominant. And with the PBMC cells, we can see humoral response. We identified in common genes, the nasopharyngeal, while same genes in the PBMC, they were downregulated. Of more interest is actually these three genes that were upregulated across the sample groups. And this one gene, which is heat shock protein A1B, that was downregulated. Our research provides insight into transcriptomic data that can be used to address site-specific differential gene expression in response to SARS coronavirus 2 infection. We've also been able to identify biomarkers that can be used and be explored as diagnostics and also be used as potential therapeutics. As well, there's need for longitudinal screening to assess the level of the potential biomarkers across the population, especially following a clinical cure. In conclusion, we have been able to identify a site-specific immune response, and predominantly in the PBMCs, we have humoral-mediated immune response. Sorry for that, I was muted by the host. So while in the nasopharyngeal, we have cellular mediated immune response. I would like to acknowledge the following, including our mentor, Sarah Karl. Thank you very much, our audience, for listening. It's time for questions. Thank you.
Yes, thank you for that presentation. I must apologize for the noise. I want to hear from the judges. Should Genomics 2 represent, or we can go on with their current presentation? Ole, can you unmute the judges?
Yeah, I just unmuted him, actually.
Okay. Should they represent starting from their pitch so that I think every group should have a leveled ground? Should we?
And if they're able to provide the pitch, that would be good to see, but the presentation was excellent. So,
Okay.
I don't,
Okay.
unless it adds considerably, I think that this team's done it was a very creative approach. So it was good to see.
Okay. Mr. Gabriel? Mr. Gabriel?
He's actually on mute, so I don't know.
Okay. Let's go on with what Alice just said. We'll play the pitch so that everybody will has. Okay, sir.
Yeah, I think we should allow them to play their pitch, please. Yeah.
Okay. So please play the pitch.
Okay, I'll play from my end.
Hello.
Okay.
We're the Team Genomics 2. Hello. We are the Team Genomics 2, and we want to tell you about our research project entitled Transcriptomic Dysregulations Associated with SARS-CoV-2 Infection.
Samples from the nose and then blood, and they analyzed.
Wale, can you continue with the pitch?
Response to COVID-19, that is, cellular-mediated in nasopharyngeal samples and humoral-mediated in PBMCs, evidencing an immunological signature. We found altered numbers of genes that could act as potential biomarkers of the disease progression, represented by increased CXCL13 GABA in Phytin3 and decreased HSPA1b in COVID-19 subjects. With our results, we were able to contribute with the knowledge in the human response in COVID-19. This understanding may support future studies in finding new therapeutic approaches to treat the disease. Thank you for watching. If you have any questions, please visit our website. We'll be happy to talk to you.
I think that's fair enough. We'll move to question and answer now from the judges. Question and answer from the judges.
I think I received some responses that they couldn't hear the audio.
Okay. Should we play it one more time, or we should just go ahead with what we have?
I think let's play it again briefly. Let's just play it. I'm very sorry for this
technical.
We're the Team Genomics 2, and we want to tell you about our research project entitled Transcriptomic Dysregulations Associated with SARS-CoV-2 Infection in Human Nasopharyngeal and Peripheral Blood Mononuclear Cells, developed during HackBio internship. This work addresses transcriptomic signature that works similar as a fingerprint. associated with SARS-CoV-2 infection in samples of nasopharyngeal swabs and cells isolated from blood that we will call as PBMCs. We are trying to find out how human nasopharyngeal cells and PBMCs react to the viral infection at the genomic level by analyzing differential gene expression data. We retrieved gene expression datasets publicly available and performed differential gene expression analysis using the seq2 package in R, and finally, enrichment analysis of differentially expressed genes between COVID-19 subjects and healthy controls. The transcriptomic profiling reveals a site-specific immune response in COVID-19, that is, cellular-mediated in nasopharyngeal samples and homoda-mediated in PBMCs, evidencing an immunological signature. We found altered numbers of genes that could act as potential biomarkers of the disease progression, represented by increased CXCL13 GABA and FITM3 and decreased age.
So we are so sorry for that. I think we can move to question and answer now. Question and answer from the judges. Questions from the judges, rather.
Okay. Thank you so much. Can you hear me? Am I audible enough?
Yes, you are.
Okay. Wonderful presentation from Team Genome 2. I just want to ask, the COVID-19 subject you use, of which descent are they? African, European? Where do you get the data from? Do we have fair representation across the continent in the data that you use for this analysis? Thank you.
Winifred, you are mute.
Thank you very much for that. Yeah, the samples were not representative across the group. So this data is only available for Atlanta. So we didn't have data for Africa. Yeah. And as well, USA. So yeah.
Okay, yeah, that's fine. I was more concerned about, when you mentioned possible application. So I'm already thinking of how the continent I represent can benefit from it. But that's fine. I think you have done good work within the resources available. Thank you.
Alice? Okay. I think she muted herself.
Please unmute her.
And there was no questions for me on this presentation. I thought it was explained very well.
Okay.
Excellent job.
Yeah, that's good to hear.
Genomics 2, I think you guys have done perfectly well. Despite the technical problems, you have done really, really well. And nice job, great work. So we'll move on to Genomics 1.
Okay, I'll be sharing the screen right away.
Okay.
So So, and I believe your presenter is ready. What? Uh, Shifumi, right? Okay, yeah. Can you see the screen?
Yes, we can.
Okay.
Remember that sad day when the Italian city of Bergamo was overrun by coronavirus deaths? It was suggested this was because of the high elderly population in Italy. But as the disease spread, deaths had been noticed in different age groups. That's why Genomics One team, through the HackBio internship, had thought to assess the COVID-19 mortality risk among various age groups using phylogenetic analysis. The Global Initiative on Sharing All Influenza Data, abbreviated by GSAD, is a primary source of genomic sequences of SARS-CoV-2, from which we had retrieved our research dataset. For phylogenetic analysis, we used some data tools shown here to construct a phylogenetic tree, helping us to understand evolutionary changes in the virus. This is a circular view of the generated tree. It holds a lot of information. We had to zoom in to find relationships between age with mortality risk and virus clade. We chose two clusters of samples from same country, one from India showing low risk with clade GH, and the other one from Singapore showing high risk with clade O, but in both cases there wasn't any certain age group relationship. For mortality risk assessment, we created an app for odds ratio calculation for the GSAT data. This app measured the odds of a person at a particular age to be at a high risk. These are the graphs of measuring the odds of a high risk, showing that as age increases, the risk increases, and the risk rate is higher in ages above 50 years. To conclude, more data sets need to be studied to understand more about mortality risk in different age groups. From all the genomics team, we all say thank you.
Okay. So you can start your presentation. I've unmuted the speaker.
Good evening, everyone. I hope we can hear me.
Yeah, we can hear you.
Okay. I am Shodunke Oluwashifumi, and I'm representing Genomics One. The topic of our presentation is COVID-19 mortality risk assessment among various age groups using phylogenetic analysis. It is no news that there is a novel coronavirus in town, and this virus, which originated in 2019, has led to more deaths than the coronaviruses that originated in 2002 and 2012. Patient mortality and the disease severity of this virus varies based on a lot of things, a lot of factors, and these factors includes age, it includes comorbidities, geographical locations, and so many others. In this study, we've been able to construct a maximum likelihood phylogenetic tree. We've also been able to carry out mortality assessments of different age groups. Our study is significant because it shows us the relationship between age, clade, and risk, and it would also assist global health organizations in developing future public health policies, making informed choices, and determining appropriate management regimes for patients. And our study also provides background knowledge on various viral clades that are associated with high mortality rates, and that will aid vaccine development. This particular slide shows all the methods we carried out to get our results. The first thing we did was to download data from GSED's data. Sorry about that. We downloaded data from GSED's database, and we downloaded a total of 4,592 genomes. After downloading this data, we had to filter out some data sets. We filtered out data sets with missing patient status and patient age. We made use of Python and BioPython to carry that out. After filtering, the genomic data reduced to a total number of 3,608 genomes. On one hand, we were able to carry out our odds ratio calculation. We calculated the odds ratio of being at high risk at a particular age, and we used a a app that we created. We created an app using EPR and EPI2 packages. We also calculated the case fatality rate. On the other hand, we were also able to carry out multiple sequence alignment using maps. We constructed a phylogenetic tree and we visualized our tree. And this led to a lot of results that we used in our inferences and discussions. This particular results, this particular slide, it shows the results for our case fatality ratio. In this particular case fatality ratio, we see that patients between the ages 1 to 40 have low case fatality rates compared to patients that are higher than that age. We see that patients at 91 to 100 have the highest case fatality rate. But because the WHO says that case fatality rates calculated during a pandemic is limited, we move further to calculate our odds ratio. The odds ratio shows that patients between the ages of 1 and 10 have the lowest risk, and patients above 81 years of age have the highest risk. Patients between ages 81 to 90 have the highest risk. And this is also in line with an Italian study that was conducted earlier this year that says that patients above the age of 80 have higher risk when compared to patients below that age. The next slide, which is showing now, shows our odds ratio of being at high risk with a given clade. Every of the purple dots shows the odds ratio for each of the clades. From this particular graph, we can see that clade L has the highest odds ratio and clade showing high risk, and clade GR has the lowest odds ratio showing low risk. We can see that clade L, clade O, clade G show high risk, clade GH, clade GR shows low risk. Now, in this slide, this particular circular diagram shows our phylogenetic tree, but we cannot make inferences without zooming into the tree because it's a very large tree. And from zooming in at this point, we're able to show a cluster of samples from India. This cluster, this triangle represents India according to our code. And sorry about that. This triangle represents India according to our code. And we see that the samples from India have a similar color in the clade section, which is a blue color, characterizing clade GH, which stands for low risk. We see that from this particular sample that at different ages, across various ages, all the samples within this cluster, except one, shows low risk because clade GH stands for low risk. In the next slide, we are able to see another section of our tree representing Singaporean samples. These samples from Singapore show high risk with clade O. The purple color here represents Singapore, while this pink represents clade O. We see that across various ages in this particular place, that patients across various ages were associated with high risk in this particular slide. Now, it is interesting to note that beyond age, there are other factors that affect the risk of a particular patient when COVID-19 is being talked about. These factors include host, patient, the pathogen, and the environment. But from our results, we could see that case fatality rate increases with an increase in age. Also, the odds ratio showed that patients above the age of 80 are at high risk when compared to patients below that age. Our odds ratio also showed us that the clades G, L, and O typifies high mortality risk. But in deviation, when we look at the Indian and Singaporean clusters, which I showed earlier, we see that though clade GH stands for low risk, it's also corresponded to low risk within various age groups. And also in the Singaporean clusters, though clade O stands for high risk, it corresponds to high risk across various age groups. In conclusion, it is true that there is an association between age and COVID-19 mortality as we have identified, but there are deviations from this knowledge as we have also observed in certain countries. Hence, the claim still requires more scientific evidence. Additionally, clades G, clade L, and clade O were associated with high mortality risk across different geographical locations and can be targets in vaccine production. These are the references for our slide. And finally, we want to say thank you to HackBio for the opportunity, and thank you to all the partners of HackBio. We also appreciate our mentor in person of Sarah Carl. And from every member of Genomics One team, we say thank you. Thank you very much.
Okay, thank you for that beautiful presentation. It's now time for questions. Over to the judges.
Hi, this is Alice. Did you look at any US data in relation to the risk that you've identified? Because, as an American, when I look at Singapore, I think of, we've fared so much less, we've done so much worse. So I'm wondering how your work relates to the outcome in the United States. If you have any ideas on that.
Our data contains, and we didn't, sorry, please, Ma, can you come again?
Oh, did you look at any US population data out of that when you looked at the different populations, did you notice any patterns of US versus Singapore?
We didn't look into US versus Singapore, Ma.
Yeah, I was just wondering if your findings, if they held up when viewed specifically towards the population in the US that's done so poorly, if you had any ideas about that.
Not at all, Ma. But our phylogenetics.
That's okay. I was just curious if anything came up.
All right.
Thank you.
Over to you, Mr. Gabriel.
Yeah, well done, Genomic Team 1. Yeah, from your findings, you did mention that the age group between 1 to 10 seems to be at low risk with associated with mortality. There's current argument in Nigeria as to whether schools should open, most especially the way we are easing down the lockdown here. The argument is let's allow the tertiary institution to open first while we keep the children at home. What difference do you think your study will make to the current argument in reopening school? Do you see any direct application to this?
Yes, our study would enlighten the government more that though the virus is out there and people can contract the virus, but children within the ages of 1 to 10, children in secondary schools and primary schools aren't at high risk compared to people of older ages.
So we can open our schools, we can allow the children.
I think the major reason they are not opening schools is because they believe that some of these children, most especially children in primary schools, have lower knowledge and they can also contract the virus from their classmates and spread it to people at home, and people at home spread it to other people in the community. So I think it's more about spreading the virus than the risk for the children.
Okay, yeah, thank you.
Thank you so much, group Genomics 1, for that wonderful presentation. Can we have a proteomics group